We have finished discussing an overview of the features that lymphocytes, both B cells and T cells, possess in the first two recorded sessions for the current module. So, this last recorded session is dedicated to a more thorough discussion on how lymphocytes are produced and the molecular basis or the molecular events that are involved in the activation of these two types of lymphocytes. So, let's start this session with T cells. Now, let's begin discussing T cell development. So, as you all know, by now, no, we were already aware of some of the fundamental characteristics of T cells. So, the precursors of T cells, the thymocytes, progenitors, they are originally produced in our bone marrow. And they migrate to the thymus in order for them to complete their maturation process, with its major function, which is to cater for T cell maturation. We can say that our thymus is a primary lymphoid organ. Now, it does not receive lymph from the other tissues, and it's actually not involved in the process of lymphocyte recirculation. So the thymus, we know it's highly active during our younger years, and eventually, as we grow old, no, the organ will actually undergo atrophy. So it will decrease in size as we grow, as we age. No, now before we begin with the process of T cell maturation, now where we are going to discuss heavily on the cell surface molecules, the selection screening process. Now, I want us to review first the organization of the thymus because you know how the cells are distributed in the thymus as a primary lymphoid organ is actually associated with the processes that are involved in T cell development. No? So across the various thymus possessing animals, actually the organ, the thymus organ, is one of the most consistent ones. So it's in terms of anatomy, in terms of histology. No, so for vertebrates that possess thymus, it's really a consistent organ. It's divided into morphologically distinct parts or regions, the cortex and the medulla, as you can appreciate here. And between the cortex and the medulla, there is a highly vascularized area known as the corticomedullary junction or the corticomedullary zone. So, aside from the regions that I think, or I'm sure we've mentioned already in our earlier, earlier modules. Also, there is the capsule surrounding the thymus. Okay, so the capsule serves as a connective tissue material that surrounds the entirety of the thymus, actually surrounding the lobes, no, produced within the thymus. And in most regions, or in most animals rather, we expect that the capsule will actually penetrate deep the thymus organ, forming what we call septa. And that septa, as we can appreciate in this picture, subdivides the thymus into lobules that are actually interconnected. And the sizes of the lobules, they may vary in terms of size, okay, and in terms of orientation, really relative to the central area of the organ. No? Now, if you're going to take a look at the bulk of the thymus, no, it's actually made up of, or most of the thymus tissue is made up of epithelial cells. Okay, so you can see there's several types of epithelial cells that we can appreciate in this diagram. No, so the epithelial cells, they form an open network, an open framework for um, for the thymus, where such framework will serve as a scaffold for T lymphocytes, for smaller populations of B lymphocytes, and then some plasma cells, as well as other immune cells. No, Now, um, these epithelial cells actually they have a variety of no of types. So, but to be specific, we qualify them as epithelio reticular cells. No, if we're going to examine the capsule or the lobular area, one lobe area of the thymus, as we can see here, the epithelial cells, they form a lining in the subcapsular region of the thymus. Such epithelial cells normally are one layer to two cell layers, or one to two cell deep, no, yung kapal ng subcapsular epithelium, no. In the outer cortex and sheathing blood vessels, these epithelial cells, they have a different morphology. So mas flatten sila, they are thin and sheet like, no. So as you can see here, let's examine here, like um, um, uh, okay, so here we have the Hassall's corpuscle. So you can see that surrounding it, no, the epithelial cells are more sheet-like, more flattened. Okay, then in other parts of the thymus, the epithelial cells may undergo or may assume a stellate appearance or star shape, as you can appreciate also in some of those that in some of the cells that are identified here near the medulla. The epithelial cells assume a more stellate shape, no, a more star-like appearance, no. Now, um, the division of the epithelial cells within the thymus is actually based on several features, based on ultrastructural characteristics like the morphology, the shape, the antigenic expression of each of the subsets of epithelio reticular cells within the thymus, and then their capacity to produce or to synthesize thymic hormones, no? Thymulin, thymosin, thymopoietin, thymic humoral factor. So those thymus associated hormones, our epithelial reticular cells of different subsets actually secrete them, no? Now, immunohistochemically, epithelial cells can also be divided, no, into four subtypes. We have our subcapsular cortical, what we have already seen a while ago, or I have described a while ago. Inner cortical epithelial cells. Then we also have our, of course, medullary epithelial cells, and then the Hassall's corpuscles. No? Okay. Now, aside from the epithelial reticular, of course, we have to describe as well the highlight cells within the thymus, which are the lymphocytes. No? Let's begin with the medulla region first. So in the medulla, the cells in the actually let's I don't know, let's take a look at the gradient muna. So cells in the cortex to the medulla, histologically they will have a gradient in terms of staining. No, so the cortex will have a darkly stained area. It will appear as darkly stained, densely packed, while the medulla will have lesser of that. No, lesser of that characteristics. Okay. In the cortex, majority of the cells that are found are immature lymphocytes, no? And you can also expect to see transitional macrophages that are beginning to be phagocytic, no? Now, if around the cortex, no, or just beneath the capsule, the subcapsule layer, we expect mitotically active lymphoblasts to be present. We cannot really see, or this figure does not represent that, or does not pinpoint such mitotically active lymphoblasts. These actively dividing lymphoblasts, no, in terms of morphology, they have round to oval nucleus with one or two nuclei, and they're very relatively they're abundant, no, and they have strong basophilic staining. Okay. Now, if you take a look at the, or if you go down, no, so from subcapsule going down, in the corticomedullary region, okay, there are mitotically active cells still of the lymphocyte lineage, but their size is less na, mas maliit na, no? Now, the corticomedullary junction, aside from containing those transitional cells from immature to mature ones, are is actually predominantly populated by blood vessels. Normally, marami mga arterioles, mga small arteries, and the connective tissue within the corticomedullary region is very scant or very scarce compared to the outermost covering, the cortex and the subcapsule. No? Now, also we can appreciate dendritic cells and a variable number of perivascular B lymphocytes and plasma cells that are present in this transitional zone. No? So they're already lymphocytes that are beginning or almost ready for circulation, for immunosurveillance, and for seeding the peripheral or the secondary lymphoid organs. So, now, of course, no, as we proceed from the court from outermost to innermost, from the superficial to the central, sa medulla naman, in the medulla, okay, we can categorize, or we can describe medulla as a funnel, no, para siyang, not funnel really, but more of a connecting avenue, no, it serves like it's like a grand avenue where your lobules, your thymic lobes, they all converge to the medulla. And then in terms of staining, the medulla is paler compared to the cortex subcortex. It's paler in staining. It's less densely populated by cells. Okay, and then majority of what we can see in the medulla are the mature T cells. There are prominent epithelial cells of the stellate appearance. The Hassall's cells. There's also a mix of monocyte of macrophages, dendritic cells, B lymphocytes. Okay, so you expect sa medulla more of mga terminal specialized cells, no, of the immune system can be found in the medulla, because at this area, this region of the thymus, they're now ready to be shipped to seeding the primary, or the primary secondary lymphoid organs and tissues, or ano na sila, no, they're ready for circulation in the blood. Okay. Here, so just to summarize everything that I've been telling you a while ago, so in terms of region, so from superficial to the central portion of the thymus, yeah, we have the capsule, the cortical region, the corticomedullary junction, and the medulla. And then the cells that we expect to see, in a way, we describe them from superficial to central. So the epithelial cells have different types, okay, based on where you can find them. Okay, from just below the capsule for subcapsular cortical, the inner cortical, the medullary epithelial cells, transitioning from the sheet-like to more voluminous, more star-like in shape, and then the Hassall's cells. Then, of course, the lymphocytes, you expect them to be mature as you proceed from cortex to medulla, immature lymphocytes, a combination of other immune cells, phagocytic macrophages, dividing lymphoblasts, mature lymphocytes, dendritic cells, our APCs, B cells that are surrounding blood vessels, and then the plasma cells, of course. Okay. Now, for your review, I have attached here some histological, I don't know, what do you call these? Histological sections, okay, of the thymus. Right. So here you can see a longitudinal section um, of our thymos. So you can see na each lobule will have a specific, ano, no, specific. If you cut it longitudinally, you can appreciate the regional subdivisions. No? Okay, so you can see darkly staining, cortical area, capsule, subcortical, transitional zone between the medullary and the cortical. Okay, also here, if we blow up a section of that, you can appreciate further. Okay, so the cortex region, the capsular region, the medullary region, you can see a lot of ano na, no, mga terminally differentiated immune cells. Hassell's corpuscles, okay? So here, you can appreciate another structure. Okay, so this is something that I will leave to you because of course, no, and since this is a recorded session, I'll in order for us to really somehow ensure that you will be able to listen to the session or watch it. No? Okay, so this is something that I adopted from this link. So if you have a guess, then you can go to this link. So let's see if your guess is actually correct. All right. So now that we have appreciated how T cells are distributed along with other immune cells in the different regions of the thymus, we're now prepared to take a look at the process of T cell maturation. So, T cell maturation involves the production of several types of cell surface molecules within a 3-week period. So the length of the whole maturation process is actually 3 weeks, and then it involves the production or regulation of the expression of cell surface molecules. So when I say regulation, kasi the process is ano siya may fino-follow na proper combination or correct combination of cell surface molecules as your T cells mature from the progenitors up to the T cell that has a T cell receptor or a T cell that can be further specialized by the action of cytokines and other antigen presenting cells, diba? You remember there are several subsets of T cells, no? Hindi lang yung ating CTLs, ang ating cytotoxic T cells at yung ating helper T cells. We also have other more specialized effector T cell types. So, as a screening process, T cell maturation will result to essentially the production of our T cells with functional TCRs, no? T cell receptors. Those T cells that do not have TCRs that are useful for the animal or to the animal, of course, they are of no potential value, and the body will delete them. The body will eliminate them. Okay. So let's begin. We were going to take a look at T cell development via stages. No? So, and each stage there are hallmark characteristics or hallmark episodes that I want you to remember. No? Because this is very basic. Kasi if you go to medical school already, this is something that you won't review anymore. So in your undergrad, it's important that you take note of these basic stuff, no? These basic narratives. No? So the initial stage of thymocyte development, here we expect that the precursor cells from the bone marrow, they will now enter the thymus, okay, and they will differentiate into what we call an early thymocyte. So the early thymocyte, if we are to describe it, no, early thymocytes, they do not possess any cell surface marker. Okay, so they're like a blank canvas. Early thymocytes are like blank canvases. Okay, they lack the CD4 and CD8 surface molecules. Therefore, they're actually given the name double negative thymocytes because they don't have that specific CD4, CD8 that we associate with with helper T cells and with our CTLs, our cytotoxic T cells, diba? So if we take a look at the cellular ontogeny blood and immune cells, diba? We can appreciate some of the cell surface molecules aside from itong CD4 and CD8. No? Let's review some. One of the by the way, no, CD refers to cluster of differentiation. So in a way, if you will encounter other CDs in the future pertinent to infection biology or immunology, so this is something that is very important for us to have a fundamental grasp of. Kasi these cell surface molecules, they give character to our immune cells. Aside from actually to very generic in description, but these cell surface molecules give our immune cells an identity that will actually dictate how the immune cells will interact with how such immune cells will interact with other effectors, with other cellular humoral effectors of an immune response. No? Okay, so it's important that we really take a look at these cell surface molecules. So some examples. Let's have a review as we relate to T cell maturation. So here, one CD molecule is CD34. CD34 is an important marker for our hematopoietic stem cell from the bone marrow. And if you notice, as you have as more commitment or dedication is acquired, or more specialization and more specialization, more restriction, less potency. Okay, so those are our three events as we progress to cell maturation. Three events you have to remember: less potency, more specialization, more restriction, okay, nawawala si CD34. It will not be associated to your more specialized types of immune cell progenitors. Okay. Another, aside from CD34, is CD45, which is present in all differentiated stem cells, such as lymphocytes, granulocytes, monocytes. Okay, so CD45 differentiated stem cells differentiated stem cells from our hematopoietic stem cell. Okay, so in terms of their nature, these cell surface molecules are actually transmembrane proteins, no? So para silang similar with your immunoglobulin, no, with your T cell receptor. Okay. Now, going back to the development of early thymocytes, no? So, let's begin our discussion with the genes that are coding for the T cell receptor. Diba? So we know that T cells are characterized by the nature of their TCRs, their T cell receptors. That the T cell genes that are encoding for the T cell receptors in the early thymocyte, okay, still we can expect that the configuration of such genes coding for the TCR component is still in the germline status. Okay? And in such state, it is expected to transition as further cellular differentiation. Of course, there's a transition to germline status to a more a status that associates with T cells, with mature or maturing T cells. In the presence of IL-7, early thymocytes will continue to proliferate in the outer thymic cortex. So, anatomically, we expect early thymocytes, diba, just below the capsule, just below the subcapsular area, within the cortical area, periphery, no? Kumbaga. So, we expect immature thymocytes or early thymocytes in the periphery of our thymus. Okay, so with IL-7, okay, these double negative cells, they will proliferate, they will divide. Okay, the next stage involves the expression of course, now yung TCR natin, which the T cell receptors, the heterodimeric TCRs, that are composed of the highly variable alpha and beta chains. And in addition, aside from, of course, the production of the unique alpha beta chains of the TCR, the expression of our cell surface molecules, our CD4 and our CD8. No? Which differentiate our CTLs, or cytotoxic T cells, and our from our helper T cells. No? T cell receptors, they are structurally similar to your immunoglobulins, no? And in invertebrates, they are coded by homologous genes. Okay, they are assembled. The uniqueness of TCRs is actually a byproduct of a process known as somatic recombination or somatic rearrangement, where the gene segments coding for the different components of the alpha and beta chains, so the TCR, they follow certain reorganization, no? So they come up with the mRNA transcript. You expect that the components of our T cell receptors, both for the alpha and the beta chain coders, magbabago-bago, magre-reorganize. No? That's why we expect a certain degree of specificity of TCRs for antigens. So the diversity is primarily, similar din naman in terms of degree of magnitude sa immunoglobulins natin. Okay, so the T cell receptor loci, they have roughly the same V gene segments, both no, A, alpha, and beta, and also their J gene segments are actually greater in number compared to the V genes. Also, what else? Okay, so I mentioned this already. Okay, so yeah. Okay, now, um, okay, so let's take a look. In terms of, or let's factor in yung arrangement nila, or yung spatial relationship with the thymic regions. We can definitely appreciate that there is, or we can definitely see that there is a regional distribution of cells expressing the cell surface molecules as you proceed from the early thymocyte stage to the stage of the stage where there is an initiation of the somatic rearrangement, and then the expression of CD4 and CD8. No? So immature double negative thymocytes, okay? So we can expect cortical area. Okay? And then as they mature, okay, you can see that they will, specialize further, eventually becoming effector cells, no? Effector T cells, either helper or cytotoxic, or further subsets depending on activation by other cells or by chemokines. No? So from capsule to medulla, from outer cortex into the medulla with blood vessels, we expect that there's a maturation gradient. No? Okay? So after maturation, we know na ready na sila for shipping. Now let's take a look at each process that's involved in this second stage of thymocyte development that involves alpha beta chain TCR expression and cell surface molecule expression. Okay, so first we have the expression of unique alpha beta chains of the TCR. No? The expression of these chains is dependent on a process called somatic recombination, where genes coding for the different genes, where the gene segments, no, or the loci, coding for alpha and beta chains, yung kanilang genes on each loci, the different genes, they are rearranged. No, these genes, they encode for the variable regions of the chains, no, which is analogous to the variable region of the heavy and light chains ng ating antibodies, ng ating immunoglobulins. Okay. So let's have an illustration. Okay, so here, so the arrangement of the gene segments resembles that of the immunoglobulin loci with the heavy and light chain genes. No, would separate variable V, diversity D. Okay, and then we also have our joining J, and then the C genes, the constant segment genes. No? Okay, so here we can see both loci, the alpha chain loci and the or the alpha chain locus and the beta chain locus. The TCR alpha chain locus found on chromosome number 14 is consisting of about 70 to 80 variable alpha gene segments, and each of them is preceded by an exon encoding the leader sequence. Um, in terms of functionality, we don't really know no, kung ilan sa variable gene segments na yan ang talagang functional. Now, in terms of the joining, the joining gene segments, letter J, okay, about 61 clusters. Okay, they are located at considerable distance from the variable segment. Okay, and then the joining segment is followed by the constant gene segment, which contains exons for the constant and the hinge domains of our chain. For the TCR beta chain, the locus is found in chromosome number 7. It has a different organization with a cluster of about 52 variable beta gene segments located distantly from two separate clusters, each containing a single diversity gene segment. No? And for the J segment, about 6 and 7, okay, beta joining joining beta gene segments. And then you can see also the constant gene segments associated with the beta chain, okay, or with the beta chain loci. Okay, the TCR alpha and beta chain genes, they're composed of discrete segments, as we have already reviewed. No? Now, functional alpha and beta chain genes, they are generated in the same way that immunoglobulin genes are for the heavy and the light. For the alpha chain, which is presented here on the upper, excuse me, the upper portion of the figure, for the alpha chain, okay, a variable alpha gene segment rearranges to a joining alpha gene segment to create a functional variable region exon. Okay, so here it's represented here. Na you can see that the intervening sequences that are not really involved or that are not really going to be transcribed or not really important for the transcript will be excised. No? Matatanggal sila. Okay? Transcription and splicing of the uh, variable joining exon to the constant alpha segment would generate the mRNA that is translated to yield the T cell receptor alpha chain protein. So here you can see. Eventually, no, magkakadikit yung in-expect natin na variable, joining, and the constant segments of the alpha chain as the mRNA is translated, okay? Or as before transcription, we expect a transcript will produce the alpha chain in this configuration, okay? Now, for the beta chain, okay, here, the lower part of our figure, like the immunoglobulin heavy chain, the variable domain is encoded in three segments, not only in the variable area. So, the variable component, the one represented here, ito, okay, for our beta chain, is encoded not only of the variable beta segment, but also by the D beta segment and our J beta segment. Okay, so you can see here. Right? So rearrangement of these gene segments generates what we call a functional V, D, J beta V region. Okay, and that exon, no, it's now ready to be transcribed and spliced so that it can join to the constant gene segment, constant beta gene segment, resulting to an mRNA that, once translated, will produce our mature beta cell. Okay, now, right. So we're good na, no, with somatic rearrangement. So yung little commentary or something that you have to remember. Okay, so the process, you remember, no, yung variable components, no, they may have the sequence of the nucleotides may be variable, depending on the batch of T cells that are undergoing maturation and selection. So we expect really a high degree of variability. Kaya we have diverse TCRs capable of knowing or recognizing different antigens. Okay. Now let us take a look at the mature TCR once that it is bound on the cell membrane of a T cell. Okay, so here, so the mature T cell receptor complex is actually composed not only of the TCR possessing the alpha and the beta chains, but also by our CDR3 proteins. No? CDR3 proteins. The complex of TCR and CD3, no, it's this whole unit here. It's one whole unit responsible for signal transduction. Okay. We'll talk about signal transduction or yung T cell activation in a little while. Actually, after we proceed with or after we finish the development lessons or the lecture on the development of both T cells and B cells. Okay, so as discussed in TCR production, no, as already discussed, we know that TCR production will require variable regions that are rearranged by selecting gene segments and deleting others to make different combinations, different possible combinations. So, isipin nyo lang, you have like 71, 80 variable gene segments for the alpha loci alone. And then we have 60 plus for the beta. So, imagine the different permutations or combinations of those gene segments, no? With of course the other components, the diversity, joining, and the constant. In terms of the order of chain rearrangement pala, no, the beta chain rearranges first before the alpha chain. So that's something you have to remember. So there's a sequence in terms of chain rearrangement. Along with the expression of the TCR chains, no, we have, of course, the production and the mounting of our cell surface molecules, our CD4 and our CD8, on the surface of our maturing thymocyte, of our maturing T cell. The appearance of a functional beta chain on the cell surface will send a signal to suppress any further beta chain rearrangement, and such signaling by the beta chain will actually trigger the thymocyte to start expressing both CD4 and CD8, which will result to the formation of now what we call a double positive thymocyte. No? So double positive thymocyte. Kasi this time, yung thymocyte natin now possesses the cell surface molecules, CD4 and CD8, along with a beta chain. Now, these double positive thymocytes, no, they will divide and they will begin to rearrange genes that are now coding for the alpha chain. Okay? So here, again, let's factor in yung spatial relationship with the thymos regions. Okay? So our immature double positive thymocytes possessing both CD4 and CD8, okay? Transitional area, cortex and medulla, so essentially parang cortico-medullary region. Okay? Now, in the third stage of thymocyte development, screening for the double positive thymocytes will actually occur. Okay, so screening. So this part or this stage of thymocyte development is very important for the screening of functional, potentially beneficial thymocytes, and the deletion or elimination of thymocytes that are of no use to the animal body. No? So during the selection process, many events, no, actually happen. So some of these events include deletion of double positive thymocytes that express potentially autoreactive TCRs, meaning TCRs that react against self-antigens, diba? Also, the screening process can result to the deletion of double positive thymocytes that express potentially non-beneficial TCRs that are not really, not really doing any recognition, or TCRs that are not, are not reactive. Then we also have the goal of the screening for double positive thymocytes, which is for the body to be able to select and clonally expand double positive thymocytes that are potentially beneficial, or that are possessing potentially beneficial T cell receptors. Now, you might be asking the question, how does the screening process happen? How does it occur? No? A process known as tolerance induction enables T cell screening. Okay, so you remember how antigen presenting cells are involved, diba, in the process of antibody production and recognition of foreign material, of foreign antigen. No? The same cells are actually involved in the process. The dendritic cells, they act as antigen presenting cells that will display self-peptides, peptides of the animal body, that are bound to MHC complexes within the thymus. So the antigen presenting cell, the dendritic cells, will do that. No? Thymocytes that express TCR specific for self-antigens, okay, they will recognize such self-peptide MHC complexes. And such autoreactive TCRs are non-beneficial, therefore they must be deleted. No? For screening of potentially useful ones, kasi syempre yung natin, no, yung mga autoantigens natin, they can really be recognized by the animal body. So we expect it's a fundamental process na that process is really going to be very active compared to the other events involved in the selection process. So for the screening of potentially useful T cells with functional TCRs, a phenomenon called interactive avidity will serve as a basis. No? So yung degree of affinity of our TCR with certain antigens, no, will be the basis. Okay, let's take a look. That. So the thymocyte screening process, no, will involve, in terms of stages, will actually have two components. No? So there is what we call an initial stage, okay, an initial screening. Sorry. Yeah, here. So we have an initial screening, okay, where your thymocyte will sorry, will interact with your other thymic cortical cells, yung mga dendritic cells natin that present the self-peptides, MHC complex on their cell surface to these thymocytes. And then our second screening, well, the thymocytes will interact with yung mga cells na sa medullary region that that are also expected to have the same capacity. No? So what happens next as the screening progresses? Kasi you expect, no, from the initial screening in the cortical area to secondary screening, you're eliminating cells that, or you're eliminating thymocytes that are not useful. So what happens next, no, is that TCRs binding to self-peptide MHC complexes, they will be tagged as having either a high avidity or a different degree of avidity. No? And those that have that are tagged to possess high avidity, okay, they can potentially cause autoimmune conditions, and therefore our body must delete them. And the deletion of such autoimmune or such autoreactive, no, TCRs, the deletion process is active, death by apoptosis. So apoptosis will be triggered in these T cells with autoreactive TCRs. No? They are what we call negatively selected sila. No? So negatively selected sila. The deletion of these double positive thymocytes expressing what we call autoreactive TCRs. No? TCRs naman that bind to self-peptide MHC complexes with negligible avidity, no? Negligible avidity, meaning hindi siya ganun ka-reactive, non-responsive. They are potentially no value to the host. No? As such, they should also be eliminated. They should also be deleted. No? So instead of spending time or spending energy, okay, and molecules to kill or to delete these double positive thymocytes that express non-beneficial TCRs, no, they're just simply neglected na hindi na sila pinoproceed or di na sila hinahayaang mag-proceed sa succeeding stages of maturation. So they're also negatively selected. Okay? Now, lastly, TCRs naman with intermediate avidity, okay, meaning they can be active in several or in a spectrum of different antigen sets or antigen types or antigen combinations. Now, these TCRs, they are stimulated to start clonal expansion. So they will be kept by the thymus, by the body, and they are cells that we can say have been positively selected. Okay, and kasi the body will expect that since they have an intermediate degree or an intermediate avidity, okay, that they might be reactive or non-reactive, or they might be able to not really reactive or non-reactive, but they may have the capacity to really respond to a foreign material. Okay. So now I mentioned though they will be clonally expanded. No? They are positively selected, stimulated to make more copies of themselves. The last stage of thymocyte development is the commitment stage, no? Lineage determination. Thymocytes that are positively selected will undergo lineage determination. Either CD4 or CD8 surface molecule will be kept, no? And either, so if CD4 will be kept, CD8 will be downregulated. So, and vice versa. Okay. So, eventually the of course the resulting product will be a single positive thymocyte expressing either CD4 or CD8. Although the process of lineage determination is almost complete, there are some instances in which some single positive thymocytes they escape the screening and selection process. No? So in such cases, their deletion still must be carried out. No? Our second negative selection will occur in the thymic medulla, where dapat nga na-expect natin mas specialized na at ready na for shipping yung mga thymocytes natin. Then eventually, thymocytes that survive this second negative selection are now processed to leave the thymus. No? These mature, naive T cells, they're now ready to seed secondary lymphoid tissues, or they can now be disposed as circulating T cells for immunosurveillance through our blood and lymphatic vessels. So in summary, T cell development, okay, In summary, T cell development results to the production of two different types of T cells, general types, effector, helper, and cytotoxic, based on CD4, CD8 absence or presence. And then the development process is considerably wasteful. So medyo very requiring siya. So vast majority of the cells that are produced in the development processes from stage 1 to stage 4, majority of them die without really being useful. And as we age, the thymus principally will undergo atrophy, no, and it will what do you call this, in our later stages. Of course, that will affect how our immunity, our immune level will actually be, will actually have certain states, no. Ultimately, positive selection works to select or to screen which thymocytes will mature. Okay? So in order to be positively selected, thymocytes will have to interact with several cell surface molecules, the MHC, in order to ensure an acceptable level of reactivity and, of course, an acceptable level of specificity. Okay, so positive selection will screen for cells with the T cell receptor that are able to bind to MHC class 1 or class 2 molecules with at least a certain weak or intermediate affinity. Okay. Uh, Okay, yeah. So, in the positive selection process will of course eliminate those that are non-beneficial because of their inability to bind or their binding with MHC self-peptide complexes. Okay. All right. So, now that we've let's see how long is this. Not sure how long this discussion is already, but since we're halfway done, no, I perhaps no, I can divide this. Actually, I was planning to really continue all throughout. In fact, tawag dito, I already recorded a session which is 2.5 hours long, and when I tried to upload it, it's kind of problematic, no, that I'm not able to upload it over the weekend. So that's why I decided to re-record and try to ano pa mas paikliin sana yung lecture, but I think we have to divide, no? I will be dividing this part 3 into 2 or 3 sessions para mas madali siyang i-upload.